So before you start recording, can you hear me? So I want
Yes,
to
I
be
can
sure.
hear you. Yeah, you can go ahead.
All right. Welcome back, everybody, and I'm happy we are here. So I took a individual opinion before people started joining, and I was speaking with Victoria earlier, and I wanted to ask again, everybody that is here, by the show of hands, I think everybody using their computer or their phones or something, even without that, if you understand 100% what happened, what was taught yesterday, please raise your hand. I would like to start with that. Everybody can raise their hands if you understand 100%. Raise your hand. Or let's do 50%. Let's start with that. If you understand 50% of what was taught yesterday, please raise your hand. So I know how to meander my way all through. I'm not going to ask you questions. So if you think when you raise your hand, I'll ask you questions. I'm not asking you. I just want to have general population and statistics of what we did yesterday. Please respond. I don't like when I'm okay, Michael. Thank you. I want to know if you understand what was taught. 50%, not everything. If you understand half of what was taught yesterday, please raise your hand. Okay, if you don't understand at all, yeah, raise your hand. If you were sleeping in class yesterday, raise your hand. Ah, why is nobody responding? Oh, thank you, Victoria. So, well, having gone through what we had gone through yesterday, she had gone very in depth and comprehensive. Okay, 30%. Thank you for responding, Jerry. So, Angela yesterday has gone through. She gave us a schematic of everything we're going to be going through in this course, and she did it in a very in-depth way, right? So I think she thinks we're all the molecular scientists at least. So I think she assumed we're all molecular scientists. So some of the things she thought was based on some assumptions. But I know the people that we have admitted, most of them are undergraduates, and Nigerian educational system, we know how it works. So I would like to reiterate and take what she took again yesterday. But I'll be writing on the things she has taught us. I'll try my best I'll try as much as possible to break it down, like to the minimum as much as possible. And I would like you to stop me if you have questions. If you have questions to ask, please stop me so that I can answer you, or I can refer you to materials. This is going to be an interactive class, by the way, so that's what I want you to know. But I would like you very much to listen. Like, I would like you to pay attention. I want everybody's attention. If you pay attention to this class, you don't have to, don't let me say this point. So if you pay attention in this class, you will not have stress when you read, because there's a material we have prepared already. Abib has prepared the material for everybody to read. That material is so interactive, it's so simple. I just finished reading it. So I want us to listen together and walk this through. So we started talking about sample to server. She started talking about sample to server yesterday. And if you know the idea of what sample to server is, it's a complete workflow. How do we even get, okay, let me start. We said we are working on genomics. We are working on sequences. Sequences of what are we working on? Where did the samples come from? What sequences are they? Where are they from? So that's the whole idea of this sample to genomics. How do those samples come to be in the first place? Where did they come from? What the person that decided to sequence them, what was the person thinking? Right? So this is how it works. When we have a question to ask, we look for the right places to ask the questions, or we look for things that could help us answer the questions, and then we go ahead and look for the answers and meander your way and just find a way to answer the questions. Of course, if it is normal academics, right, you go to Google to find materials to help you answer the questions. If it is of course, anywhere you know where to look for your answers and go get your answers. So the same thing with this. Let's assume I would ask a question, I would make a scenario. Let's assume that I want to know if if an organism, don't let me call it isolate, it's an organism has resistant genes inside of it. So I want to know, let's say a patient came to the hospital today, and I'll be using this as my example most of the time. The patient came to the hospital today, and when the patient got to the hospital, the how am I going to explain it? The lab, I mean, medical laboratory scientists were able to isolate the organism, right? And because of time, or maybe because we are very, very rich, we don't want to go through the contemporary route of doing AST. If you know what AST means, is the act of doing antimicrobial susceptibility testing. You want to know which antibiotics will work if you give the patient so that the infection will go away, right? So you are in an advanced country. You want to know which organisms will work, which antibiotics will work, which antibiotics will not work. So you send the sample to the laboratory, right? So it's a lot of things. So in before you even get in the first place, the first thing they will do is they will get to prepare media. For us here, I believe some of us will have an idea of microbiology or what's the name of plates preparation and all these things. So they'll prepare the so-called agar, depending on the micro or the organism you are dealing with. After they have prepared the agar, then we can now start culturing, and then after you culture and all the like. So all of this is what I'm going to talk about today, right? So let's go on to the. By the way, that's me. That's my face. I just wanted to show my face because why not? I can. Next slide. Now, why does the wet lab matter for bioinformatician, right? The computer is powerful, but it cannot fix poor quality data. So when you want to do your analysis is as good as the data you have, right? The analysis is just as good as the data you have in the first place. So if you have good data, your analysis will be fine. If you have bad data, your analysis will be bad. It's as simple as that. So if you have a good data that will help you find all the solutions to your problems, to help you answer the questions you have to ask, it is good and all and good, and then you continue. But if you have bad data, the results you will get will be very, very useless. So understanding what happens in the lab will help you troubleshoot errors. Let's say, oh, there is a contaminant from the bench. You will understand these terms very soon. So just hold on with me. So there's a contaminant from the bench. There is um there is a lot of things happen. There is adapter ligation. There is adapter in my sequence. There is this and that. This terminology you understand very soon. By the way, just listen to me. So if I don't know what happens on the bench as a scientist, you don't have to be on the bench with them. By the way, as a mathematician, but you should have an idea, mental map of what's happening, so that when you see results, when you see problems, you'll be able to troubleshoot and know, okay, where this is where the problem comes from. Now, to be able to know where the problem comes from, we'll go from the beginning till the end, right? How the sample is processed in the laboratory. So if you do microbiology or if you have had a microbiology class, I think you wouldn't have any problem. But if you haven't, please still listen and watch so you understand. And if you don't understand, the materials will give you make you understand. Just listen to me. If you have questions, by the way, I said it, then you can stop now. So this slide has explained it. You want to do analysis on a very high quality data. If your quality data and you do not just the quality data did not come from thin air, it came from the laboratory. So if they make a very stupid mistake in the laboratory, you are also going to inherit a very stupid data, and then the analysis will be stupid as well. So everything goes on from there. So we want to go back and see what happens in the laboratory, right? So the first thing that happens in the laboratory is they isolate identification and culture. So they want to isolate, and then they want to culture, right? So I said it earlier. The person that came to the laboratory, the person has a wound in the leg, right? And it has been infected, right? Since the organism has been infected in the hospital, the wound has been infected. You want to know what organism is there in the first place. So you have to culture the organism. So growing a pure culture from a single bacterial colony to ensure that we are sequencing new organism. By the way, before I get here, sir, the first thing they do is they will prepare media, right? And depending on the organism, this is work of scientists. So you don't have to understand all of this part, but I would be happy if you do. There are different type of media in the laboratory. There is what's the name? Acidious agars, enriched enrichment. There is what's the name? Basal medias, nutrient agar, molar-eating agar. There is selective agars. There are differential agars. So this is not your work. This is work of the scientists to know which agar to use to process the sample. So it's a one swab. Of course, you're expecting fastidious organisms. Or what about your organisms that they're expecting? So they would use a media that would help them grow that organism. So after the organism have grown, they will purify, right? So there are some, let's say it's a urine infection. Let's say somebody comes in the laboratory with a UTI, urinary tract infection. If I want to culture that organism, I know that for somebody to have an UTI, most of the case, most people that have UTI in the laboratory is just what's the name? It is just one organism that causes the problem. It's just one organism. Maybe no matter the organism, it's always just one. You cannot, it's very rare for you to find two person that has UTI. UTI means urinary tract infection. There are two organisms to find. So let's assume that this person came with wound, and we have assumed that it's one organism that's in the wound. Let's call the organism unknown. We don't know it yet, right? So they will purify the organism in the laboratory, right? They will culture the organism on agar. It will grow. They will incubate it for 24 hours. After it has grown, they purify it, because even before you do AST, before you do identification, you must have a pure colony. And this is very important for you going on with your research. So I said we're in a very, very high income country, and we don't do all these shenanigans. We just want to go ahead to identify and then to do sequencing, to do all of that things. So they culture the organism on a very nice agar selective, and then they select a very one single colony. Now, if you have been to the bench before, if you have been to the laboratory before, when you culture something, what I was saying earlier, you will see a lot of contaminants that will also grow with it. So it takes intelligence and it takes experience to be able to know, okay, this is the organism that I'm looking out for, right? Sometimes there are two organisms you're looking out for, sometimes it's one you're looking out for. So it takes experience to pick. So this is on the part, I'm telling you what scientists do on the bench now. So yeah, bioinformaticians, you are in the dry lab, so you're not in the wet lab, so you might not understand this, you might not see this part. But if you have seen it, I think it's a win-win for you too, by the way. So when I see a culture on the plate, I know that okay, this is most likely going to be the organism I'm looking for, and this is most likely going to be a contaminant. Sometimes you find pure colony at your first go, you are very lucky. So after I've gotten my pure, even if you have pure colonies at the first go, you go ahead and do your identification. If it's in a medical laboratory science lab, right? It's a lab that we work. It's a lab in UCH, for instance, or a lab in Sin Lab doing diagnostics. So there are medical laboratories. So they go ahead and identify and do AS and all those things. So we are bioinformaticians. We're in a genomic lab. So after they have identified the organism, after they have gotten a single colony, they make sure they identify the organism, right? So identification of the organism is important, because even when they want to identify the organism, they have this panel of biochemicals that they do. So it is very, very simple and straightforward. They will just run biochemical patterns. Even before it used to be hard, now they do something, they call it API. Or if you have heard of Vitec before, it's very easy. They will just run the organism on Vitec, and then Vitec does different biochemical patterns. So what that means is, if I'm looking out for Joseph, right? Let's say these people in this case, I don't know you, all of you here are bugs and whatever, but I want to look for the name of Joseph, but I know how Joseph looks like. Or I'll say that, oh, there is someone I'm looking out for in the Info UI community. He's short, he's dark, he has a MacBook, he uses this, he uses that. Maybe I can tell you 4, 5, 6, 7, 8, 9 characteristics about Joseph, right? And if I have said all of that, I think he will be the only person that has that characteristics, right? So that's what happens with biochemicals. Now imagine if I did not pick a pure colony when I'm doing my biochemical. So if you have seen a plate before, this is what I was trying to say. Some colonies, they overlap on each other. You thought you had picked a single colony when you are picking. By the way, do we know what colony is? I want a chorus answer. Unmute and tell me yes or no. Do you know what a colony is? Have you seen it? By the way, this place in okay, I know
Yeah.
that. Good, thank you. So this is a plate behind. So this is not how this is culturing for some samples. So by the way, this is not for you. In laboratory, different samples, medical laboratory specifically, different samples are processed in a different way, right? So samples like this that you see that they don't streak out, and they do this is just from my expertise. I'm just sharing my knowledge with you. That they do primary inoculum like that, they don't streak out. This is how they process eye vagina swab or ECS or eye swab. If you see eye swab, if you see eye vagina swab, if you see endocervical swab, they don't stick out. They do impressions. They call this impression smear. But that's beyond the point I was trying to talk about. So If I have prepared my colors like this, look at all of these things here. I think, can you see my mouse? Oh, you can see my mouse now, right?
Yes, we can
Yes,
see it.
you can see it.
My mouse is on the, yeah, thank you, because I can see what two people are seeing. So I know you can see my screen, my mouse. So this is the primary inoculum, right? If I want to pick a colony, look at, this is one colony here. They have different colonies here. I wouldn't pick from something like this because this is clumped together. I want to pick a single colony. I'll pick something that is just standing alone here at the side. Even with this, I'll still have to purify again because something might be under it. Now, there are different colonies here. I picked what I want to work with. I purify, I find out, I make sure that there's nothing going under it. Because if I want to identify this organism, I don't know what this organism is. And if you pick two colonies that are very side by side, let's say the two colonies are different organisms, right? If I should go ahead and do biochemical what's the name, biochemical identification on the two colonies I picked together, I thought is one, the machine will tell me poor identification or there will be an error because you told me I gave you biochemical pattern for I gave you an organism and I want you to help me find with a set of biochemicals. So I'm seeing attitude that looks like Joseph. I'm seeing physique also that looks like let's say Akintayo. So two people looking like the same way. The machine will be confused. It will tell you I don't have confidence in this identification. It will tell you poor identification. What if you see poor identification most of the time when they are trying to when you are working on the bench trying to identify an organism is most 90% of the time it is based on contamination. This is contaminant. So there is always contamination that has happened, right? So you don't want to have that even before you even start doing your before you start doing what do you call it before you start doing your identification with API or Vitec or whatever thing you want to use to do your identification. When you do your gram thing alone, I'm sure we all here should know gram thing. Abi, we don't know, we know, right? Good, thank you. So before you even do identification and you do gram stain alone, if you view it under the microscope, you would see mixed cultures. You might see a gram positive and a gram negative together, and then you know the colony you picked is not pure. So colony picking is very, very important because that's the beginning. If you should pick a very bad colony, if you should pick contaminated colony, maybe two or three colonies that are of different organisms, if you are finally sequencing and you are finally doing assembly, you will see when you start talking about fast QC, you understand these points better. You would see a lot of data that are erratic data. You will see your genome size. Let's say genome of E. coli. Let's assume I've not gotten the name of it. Let's assume the organism I'm working with that I discovered that I picked that I'm trying to pick was E. coli. E. coli is about 5.1 million bases, right? But when I was picking, I picked staph with it. So I can't remember the genome size of staph now. Maybe Joseph could help me with that. Let's assume 3 million or let's assume 10 million bases for staff. So if I should do my 5QC, I'm supposed to see a consensus of like I should see a dome shaped curve on my genome size around 5 million bases. But you will see some extra bases, some extra curves somewhere else when you start doing a 5QC, then you know that there's a problem. Something has contaminated your data. So you don't want that to happen. So before you start doing that, before you get to that phase, scrutiny is very important on the bench. So you want to avoid mixed signals. A mixed culture, it's like two people talking at once. The resulting geometric will be confusing. Jumble is difficult or impossible for you to assemble correctly. Of course, when you start assembling, when we get to the read and whatever, you understand what I mean. So to inform your strategy, knowing the organism helps you choose the right DNA extraction kit and sequencing platforms. So I've talked about purification. I didn't talk about I talked about isolate culture and isolate identification and culture now. Then we are going to move to the next part, which is DNA extraction, right? So this is typically how our labs looks like, by the way. Most labs, my lab in usage, lab in UI, everybody that does DNA extraction and anything genomics is most likely how it looks like all the time. So what is DNA extraction? You remember when you were doing your bio 1211 or bio zoo 111 or whatever zoo you did in the 100 level? I'm sure everybody on this call has done the zoo. And they will ask you to go and draw bacterial cell wall or a bacterial cell rather, or a plant cell, bacterial cell, prokaryotic cell, eukaryotic cell. Those things I used to think they were punishment because they're always drawing and submitting and they will not see our logbook anymore. But since when you start working with this and then you start understanding what it means. So what is DNA extraction? If you remember your prokaryotic cell, right? If you have drawn it before, you have seen, you know what I'm going to do. So let me just continue. Let me see if I can switch slide. You know how? Let me just continue. You know how your bacterial cell wall looks like. Your bacterial cell looks like. There is a very thick cell wall outside. They don't have cell membrane, but they have cell wall. But they have everything inside their cell wall. So it's like a big hall that you do all your activities. So all those warehouse that inside one warehouse, of course, it is very much fenced outside. But inside the warehouse, there is the person that is, let's assume it's a factory where they produce Coca-Cola. So inside one place, everybody together, person that is producing the concentrate, person that is mixing the water, person that is purifying the water, person that is using the machine, everybody's in one place together. So that's what a bacterial cell wall looks like. They don't have partitions, right? The same way our human, our characteristic cell for human cell, we have partition. There's nuclear membrane, there's a ribosome, there's a vacuum reticulum, everything has their own compartments. In bacterial cell walls, they don't have compartments. So everything is inside together. But they have something that is very strong outside. They have a very strong cell wall outside. So DNA extraction is, how do I invade this building? How do I break into the cell wall to take DNA? Mind you, they don't have cell wall, they don't have membranes that is protecting their DNA. Their DNA is just floating in the cytoplasm. So DNA purification, DNA extraction is a very, very important step. Now, let me take it back a little. I told you they are coming from cell wall, and I mentioned something earlier about gram positive and gram negative cell wall. I want to throw it out here. Of the 2-gram reactions, or of the gram positive organisms and gram negative organisms, which one has a thicker cell wall? I want everybody to answer, or put this in the chat, something. Which of the
Gram
organisms?
positive.
Good.
I'm positive.
Thank you. Have a thicker cell wall because they have a very thick peptidoglycan layer, they have teichoic acid in their cell wall. Of course, gram negative also has, but they are not as thick as gram positive. Gram negative have more of lipid, lipopolysaccharide, right? I think that's what they have more than gram positives. Imagine I want to extract an organism of getting the organism. It has a very thick cell wall because it's a gram-positive organism, and I want to take the DNA out of it. So you have to find a very suitable protocol that will help you break the cell wall, right? By the way, let me digress a little bit. If you have read about DNA extraction before, there are like 3 major ways of you breaking the cell wall so that you can get inside. You can use mechanical method. That means you're actually using hammer, like you're using hammer to break the cell wall. But instead of using hammer, they call it beads. We have beads that we use to break cell wall. And if you have been to a DNA extraction break before, you would see it. You can use enzymatic methods to break the cell wall because, of course, we know the composition of the cell wall, the glycan layer and teichoic acid. So what if I make an enzyme that can actually break the bonds that are joining each individual molecules of the glycan layer together? So that's the second one. The third one is very good and archaic. You can use it. You just boil the thing to break the cell wall breaks off, right? You boil it and boil it till the cell wall breaks off, right? Now, of these 3, some are more conservative than the other, and these things tie together. So I will jump forward, I will jump backward. So you just be with me. If I am going to be working with, let's say I want to research on, when I'm going to be sequencing, I'll be sequencing or I need long reads for my sequencing. Let's say I want to work with plasmids, or I want to work with mobile genetic elements that they fly around. So if I want to work with these things, right? I want to make sure that I don't fragment my genome so much. Because imagine, let's use the example of this warehouse I told you earlier. The DNA is floating inside the warehouse, and I want to break in to escape the DNA. If I should start using hammer on the cell wall, that's the mechanical method of extracting, of breaking down the cell wall. If I should start using hammers to break the cell wall, of course, I will get my DNA out, but my DNA will not be in the code. Who else is unmuted? Somebody else is talking. So if I use what's the name, hammer to start breaking the cell wall, to get inside to extract my DNA, I would obviously I will break the cell wall, I will get the DNA, I will get as much as possible. But what will happen is my DNA will not be a complete human being anymore. Will not be complete because I would have fragmented it with the agitation of the amas. Because DNA is fragile, by the way. Of course, it is stronger compared to the RNA, but it is still fragile. It's just a baby, right? So you want to extract your DNA. So your sequencing technology will inform the kind of extraction you want to do. Do you understand what I'm trying to say? So if you these things they will come together at the end. Just listen to just absorb it that I'm trying to take. So you want to extract your DNA, you want to use the best because you have to choose what is right for you. So the people that are this is not for you by information, but when you are designing your protocols together, you should be there because if you want something that will give you high quality data, you should be with them. Think from the end, don't think from the beginning. Think from the end. You want the data that will be able to help you get this result. So I want to work on plasmids, I want to work on integrons, I want to work with this or that. Make sure that you have something that would help you with long reads. And if you want long reads, of course, you know that you don't want to fragment your DNA. You don't want to agitate it so much that your DNA will break into pieces. So instead of you going for a kit that will help you extract by mechanical breaking of the cell wall, you go for a kit that maybe uses enzymatic. So that one is more gentle. You just pour your enzyme. But all these things they come with trade-off, right? If I'm going to be using mechanical, in 10 minutes, 12 minutes, or 15 minutes, I'm done breaking down the cell wall, I'm inside. If I'm going to be using enzyme, remember enzymes, right? They work at 37 degrees and then they work for a period of time. So I would have to pour my enzyme in my DNA or my organism mixture. Then I'll have to put this in a water bath and incubate for a long time and then wait for the enzyme to start working. And then you understand this process. So they come with a trade-off, by the way. But it's good for you to know the best extraction method for your organism, and it is based on your study. So after saying let me now read what's in my slide. So it's the process of breaking open the cell and isolating the pure genomic DNA from the cultured isolate. So use how is it done? You have to use your manual protocols and or commercial kits. So I have used Zymo, I have used Kiagin, I have used Promega. I don't know why I didn't include Promega here. So a lot of things can be used. A lot of different people are making different kits. So if you want to choose anyone for your experiment, think from the end. What am I going to look for? What am I looking out for? If I should use this kit that would do mechanical, will it? So you just have to think. If I'm going to be doing a work that would infer a short read, okay, Shegun, show the way. You can ask a question and I will continue. Somebody raised up your hand though. Shegun, show the way I had. If you are not asking your question, I'll just. Oh, okay. Let me continue then. Said it was a mistake. So if I am going to be, I lost my train of thought. By the way, if I'm going to be working, oh, I remember. If I'm going to be working with, let's say, Illumina. Illumina does short read sequencing. You know what this means later, by the way. But and you can also read it up and Google it. So I know that I can actually still use mechanical way to break my cell because, of course, you fragment it. If I do not fragment it when I'm extracting, when I start to do my library preparation, I'll still fragment my DNA. Do you understand? So that's what happens. So I said all of this because it is it's important for you to know the method of extraction of DNA so that it will in fact it will affect your DNA and whatever. By the way, one point I need to make to you that I have not made now. Well, hold on, I will stop sharing and I will also share back. I want to share my whole screen instead of just the slide so that I can go. I want to go on Google and start showing you some things. Okay, so back to my slide, right? Let me close this one. Back to my slide. So I have this here. Now I want to use Escalade Draw. Okay. Ignore this. I have drawn a lot of things. So this is the genome of the bacteria. Or let me divide that. Let me use this. No. This is the bacteria. And inside the back, you can still see my screen, right? Can we also see my screen?
Yes, yes.
Okay, good. This is the bacterial cell wall. This is the DNA inside. So let's say I fragment, oh no. After fragmenting the cell wall, I will have quite a number of things. I was looking for freehand. There used to be freehand. So I have quite a number of things inside my cytoplasm, right? And I've broken the cell wall, of course. Even so, let me sound a note, by the way. When you when you break with enzymes too, you cannot still get a complete genome. You understand? You will still have fragmented genome. So it is important for you to know these things. But it won't be as fragmented as mechanical. So you cannot, it is almost impossible for you to get a complete genome when you fragment with enzyme, even with it is almost impossible. So I would like you to take note of that. So those fragments that you have, DNA you have gotten, is what you now use to prepare your library, right? So why does this method matter? Poor extraction leads to low DNA yields and poor quality, and then contamination from samples, from reagents. I told you again, imagine the sample you use to extract your DNA is like a mixed sample. You have, instead of you to extract the DNA of an E. coli organism, you extracted E. coli with staph, right? Two organisms that are from two different generations. You would have problem when you start your sequencing because you will still extract, because your extraction, you extract the two of them together. So you can get contamination from samples, contaminated DNA samples. Your reagent can also contaminate. Let's say you are not careful enough when you're doing your last DNA extraction, and then you have contaminated the reagent with one of the DNAs that you were extracting the last time. Now you transfer that reagent to the new sample. So the sample you are working with will be contaminated. And till you finish that kit or that lot, all the extraction kits, all the extraction you will do with that kit will always be contaminated. And you will keep wondering, have all the last 6 sequencing that we have done, everything is contaminated. All the things are not passing fast QC. So you start wondering. So this is where the things you start considering when you start having problems. This is where you should start thinking. Does it mean when they extracted something with reagents has contaminated all our anal extractions from beginning to the end? Let's say the person that did the extraction did a very good did the culture and picked the isolates, did a very good job. If you have still contaminated your reagents, now wahala go see there too. So kit compatibility. Some kits are optimized for long read tech by preserving long DNA fragment. I mentioned this earlier. While some are suited for short read tech. By the way. So moving on, I think I spent too much time on this DNA extraction. Now, after I've extracted my DNA, by the way, if you have seen where they have extracted DNA before, even if you are not, just imagine. I'm sure you have seen 1.5 million pen tube before. Most of our DNAs, we extract them and we put them in a small tube. Except for some kits, let's say a kit like Promega, every other kits that have used to extract DNA, it's just water. You will see what you see is a clear water. You will not know DNA is there or not. But I'm just believing that all the process you have done in the last 4 or 5 hours or the last 3 hours, depending on the kits that you are using, has removed DNA into that tube. So you are believing, you are hoping. And you know science. Something about science, why I love science, why science is different from religion. Science is evidence-based. You don't want to tell me that I have extracted DNA and you don't give me an evidence that there's a DNA inside the sample, right? Cannot just that's Sakamanje, that's Agbo science. So you really want, sorry to people living in Agbo. So you want to know what is actually going on and not just fake it, not think it. So that's why quantification and normalization is better. So what is quantification of DNA? After you have extracted your DNA into the tube, so let's say you just finished extracting 24 samples, you just finished extracting 48 samples, depending on how strong you are, right? You want to now know, after all this, my 4 hours, do I actually have DNA in my sample, right? How do you test if you have DNA in your sample? You have these things. There are different things to measure the concentration of your sample. So accurately measuring the concentration of your extracted DNA and adjusting it to optimal amounts is required for the next step. So there are different kits that can help you quantify. And how does this kit work, by the way? Example is this for this qubit. I've used this one single qubit. I've used a qubit with 8 wells before. So if you are using qubit, what qubit does is it comes with a die, and those dies are they have affinity for DNA, right? So if you pour your die inside DNA and you measure it with the qubit, so only the dies that have found the DNA will be viewed by the machine. So if you have water and there's no DNA, the dye you have added to the sample will not bind with anything. So it will be excited when you use fluorometer. I'm just giving you a very, very simple um example of how it works. That's how it works basically. So qubit will tell you and help you. So as much dye binds to DNA is as much what's the name quantity of DNA that is there. So if you have just one DNA, just one fluoro, let's say, let me assume, I'm just using this as an example. Just one fluoro, one dye, like one piece of dye will bind. But if you have 1 million DNA segments, 1 million DNA dyes, 1 million dyes will bind to 1 million DNA, and then the fluorometer will see, okay, I've seen 1 million DNAs inside the sample. So that's how it works. So it helps you to know if you have DNA inside of you, and it helps you to quantify it. But there's a limitation to qubits, even though that's one of the most widely used quantification methods in science, right? In genomic studies. There's a limitation. The limitation is that you can only assay for DNA at a time. Do you understand? You cannot assay for you don't know if there's a contaminant in your sample. So sometimes when you're extracting your DNA, you extract other things. Your proteins, maybe when you're extracting, you didn't purify your protein very well. You didn't remove all the proteins you're supposed to. By the way, when you're extracting DNA, remember I told you, you break your cell wall. After you've broken your cell wall, remember there are other things inside the DNA. What you are trying to do in DNA extraction is clean up the DNA in such a way that every other thing that is inside that sample gets removed and you have only DNA. So you remove the proteins in the sample, you remove the RNAs in the sample, you remove every other thing that's living in the cytoplasm. Does it make sense to you? So that is what DNA entails. So if you didn't do that well, right, and you just extracted, of course, DNA will extract, but you will have other things, other impurities inside. You have RNAs inside your sample. You have you have what's name, proteins inside your sample. So if you are here quantifying, of course, the fluorometer will only bind to DNA to the DNA and give you the figure of DNA, but you will not know if you have impurities in your sample. Does it make sense? Now that's where this number two sample, number two instrument comes into play. Nanodrop. Nanodrop measures nucleic acids, everything together. By the way, this also has its own problem. RNA, DNA, but it also helps to quantify every other thing. So if there's protein, nanodrop will give you the figure. If there is so any other impurity that is in your sample, nanodrop will help you quantify that. So that's how you know if you have problem, if you have a contaminant in your sample when you have that is not DNA when you are using nanodrop. So qubit only gives you the figure. Nanodrop gives you the figure of every other thing that is inside. Doesn't make sense. Just does all the nucleic acid as one, then it does protein as one, it does other impurities as one. But does it make sense to you? So why is this not negotiable? The amount of input DNA directly correlates to the final sequencing yield. Every preparation kits have a very specific input range. Too much or too little DNA results in a field library preparation. So before I move on to the next step, we have to talk about normalization. I talked about quantification already. Now, I have extracted my DNA, right? Let's say, by the way, if you extract DNA, you don't extract from one sample, though. Of course, it's a waste of time. Imagine spending 3 hours from one sample. You can spend that same 3 hours for 24 samples. So what we do is we batch samples together. We extract 24 samples. Some people have strength. We extract 30 samples at the same time. So I think like to extract 14 samples at the same time. All the best. Or Apollo, I extract 14 samples at the same time. All the best to you people. I don't have that strength. I just have 24. So it depends on how or your instrument that you use for extraction. There are better extraction kits now, that's automatic extraction kits that can help you extract faster. So if I'm extracting 24 samples, right? After I've extracted my samples, all of them will not yield the same way. Do you understand? Some will have more yield than the other, and it is not your fault. It's from the starting quantification. If you have seen how they extract DNAs before, they used to do a preparation before you extract your DNA. So they will incubate in TSB for 24 hours, agitation, and all of this is to increase as much. So not all organisms will grow the same way. Some organisms will grow more, some organisms will grow less. And if you extract all of those together at the same time, some organisms will give you, let's say, per meal, I have 1,000 nanogram of DNA per meal. And some other extraction you get just 40 nanogram per meal. I've done some extraction that I was hot. After spending 3 hours, I have just 26 nanogram per meal. Very little, right? And I don't blame them. They are gram positives. I hate gram positives. I hate the working gram positives, by the way. So this has to do with this has to lead to normalization. So imagine if I am working with, if these 24 samples are what I want to go ahead and sequence. I have a sequence. I have a sample that has 1,000 nanogram per mil sample. I have a sample that has just 26 nanogram per mil. I want to sequence the two together at the same time. If I report the two to library preparation and sequencing at the same time, I would have problems, right? And the problem I'm going to have is that the one that is 1,000 nanogram per mil, because they all have it. If you have seen the sequencing technology before, let's assume you are using Illumina. Even if you are using nanopore, both of them, they have a flow cell that the sequencing happens. The ones that is plenty, it's like you putting what's the name? Yeah, we are putting primary 4 students with SS3 students to play at the same field. You cannot make sense of that match because the SS3 students are built giants and everything. They will dominate the field, and primary four students will not dominate the field. So you want equal leveling chance. So all the DNAs that are very, very high in concentration, you dilute them. All the DNAs that have low concentration, you concentrate down so you can whatever we ought to do, dilute up or dilute down. Either you want to so you and you want to achieve the same DNA concentration going into your library preparation, going into your sequencing. So that's the whole idea of it. So normalization is just as simple. Simple. They use is water that I use, not the water you drink in your house, by the way. We call some things nucleus free water. Water that don't have nucleus because we are working with DNA. Remember, DNAs will destroy DNA. And let me let you in on a simple thing. There's something they call normal flora in your body, by the way. So if you have normal flora in your body, an example of normal flora is Staph aureus. If you know Staph aureus, you know Staph aureus biochemical reaction, but Staph aureus produces DNAs by default. If you want to test for Staph aureus in laboratory, some standard labs they use DNAs to test for Staph aureus. It's not just Staph aureus, by the way. So if Staph aureus produces DNAs, you know, if it is in your water, and of course all our water, me, I will not like even the one everybody listen to me, you have Staph in your water at home. If you have to have DNAs, if you use it to normalize a DNA solution, that DNA will destroy all the DNA. So it's water that you are going to be sequencing. So you don't want that. You don't use your water in the house. You get to the sequencing laboratory. They have a special water that they use for sequencing. They call it a DNA-free water or RNA-free water. We don't have the type of water. So they use waters. When I say no water, I'm talking about nucleus-free water, by the way, or NFW. If I say NFW, nucleus-free water, I'm saying the same thing. So they use this to dilute down or dilute up, depending on what you want to do. And then after that, then you can now start doing your library preparation. So, having talked to this point, I want to know if you understand. Do we all understand till this phase? I'm repeating what she said yesterday, but I'm trying to break it down as much as possible so that we all get it. I want to see everybody, if you understand it, please. Okay. Thank you. Thank you. All right, I'll go back to my slide now and continue presenting. All right. Thank you. Thank you. So, slideshow. Good. After, did someone raise their hands? I heard low. All right, so Akitayo, please ask your question.
Right, sir. Good evening. Please, can you come again to that normalization concept? I don't really understand it.
Can you hear me, please?
Sir, please, can you come
I think
again?
my network
I
went
didn't
off.
hear anything. Okay.
Yeah, I think can you hear me now, everybody? If I can,
Yes.
Yes, sir.
can you hear me now, everyone?
Yes, sir. I
Yes.
can hear you now.
Okay, thank. All right. So what I was saying is this: if you extract 10 samples together, it is beyond your control to tell which one will yield more or not. So even if I take 1 ml of the same sample, let's say I have put my sample inside 1 ml of TSB and incubated it overnight and extract my DNA, some organism will grow more than the other. So if I have 1 ml of TSB, if you know TSB, triple soy broth, if I have 1 ml of TSB, I can have 1 million copies of a DNA inside one sample, inside 1 ml. But the next bottle that I'm going to extract, I'll have 1 billion copies, right? If I should extract the two samples, the one that has 1 million copies of DM, 1 million copies of organisms, second one that has 1 billion copies of organisms. If I have extract the DNA, of course, it is very, very sure that the one that has 1 billion copies will have more, what's the name? Will have more yield than the first one. So if you are quantifying the DNA at the end, the one with 1 billion samples in the first 1 billion organisms will have maybe 1,000 or 2,000 or 3,000 nanogram per mil of DNA. But the one that has 1 million might have just maybe 50, 60, 70 nanogram per mil because the starting concentration of the organisms that you're extracting from each of them are not the same. And you don't have control over this. You don't have control over this. But before you start doing your library preparation for sequencing, before you start doing all of those, you must make sure that both of them are of the same quality. Because why? We sequence this, there's something we call pooling. When I want to sequence, I don't sequence one sample, I sequence 20 samples together, I sequence 24 samples together. By the way, in nanopore, in Illumina, you can sequence 96 samples together, right? You can sequence 300. There library their barcoding kit that can help you do 300 and something samples together. That's how crazy it is, by the way. So if I want to sequence, let's say I want to sequence the two of them samples together. The first sample that has 1,000 nanograms per ml. The first sample that has Afoluwa, you are raising your hand. Okay. The first sample that has what's the name? That has 1,000,000 samples, 1,000,000 bacteria starting, and the second one that has 1,000 bacteria starting, 1 billion bacteria starting. They have different concentrations of DNA at the end of the day. You don't have control over that. But when you want to go ahead and sequence, you don't want to put the two of them equal quantity. You must make sure that there's equity, because it's only the one that has 1 billion, or let's say one. Let me go back to the concentration of the DNA, 1,000 nanogram of DNA, and the one that has what's the name, 5, 10, 26 nanogram per mil of DNA. If you put both of them on the same flow cell to sequence, the bigger ones, the bigger concentration will take more space than the smaller ones. So when you have your sequence, your read will be discrepant. I'm trying to see if I can find something to explain that, right? Hold on. Let me see here. So bacteria 1, bacteria 2. Bacteria 1 here has this is 1,000 nanogram per mil. This sample 1, by the way. And then this sample here has 50 nanogram per mil. After my extraction, after I'm done extraction, this is after I told you we quantify. When you quantify, this is the figures you get. You get figures nanogram per mil. I got 1,000 nanogram per mil for sample 1. I got 50 nanogram per mil for sample 2. If I should go ahead and sequence both of them on this flow cell, it is obvious that this one will take more space. It will take more space on the flow cell. You understand what the flow cell means very well later. Don't have to worry. This is part of the flow. So this one will take more space. Let me delete this. This one will take more space, while this one will take small space, like that. This one from here. So these two is not equal. So when you this is let's this is your sequencing machine. When you get your data, you will get more data for sample 1 than sample 2. And that is not good, because there's something they call read depth, there's something they call number of which you get, the depth of which you get will be will be in favor of this sample than this sample. You don't want that, right? So what you do is you normalize. So for you to normalize, you dilute this one down in such a way that you have equal concentration. So now when you are diluting, they will now have maybe not the same, but somewhat the same. You now have this one can now have, you will dilute it or concentrate in such a way that this one can now have, let's say, 50 nanogram per mil. So both of them have the same nanogram per mil now, and then they have the same equal chance on the flow cell, and then your reads, you get your reads, and everything happens. I hope you understand what I'm talking about.
Yes,
Do
sir. Yes,
you
sir.
understand, Akintayo?
Yes,
All
I
right,
now
thank
understand
you.
now. No, thank you.
Okay, thank you. So let's move on. So I have explained qubit, I've explained nanodrop. Tape session is also like the both of them. Very beautiful. Even if plot, you see the fragment size, it tells you when you have extracted your DNA. I told you when you are extracting DNA, especially mechanical method of cell lysis. We start with mechanical method of cell lysis. Your DNA needs to be fragmented, right? Because you are going in with full force. Even, no matter what you do, not just mechanical, you will fragment your DNA. But if you are using enzymatic and what's the name and it, you would have longer. You have somewhat longer DNA strand, because you fragment less than the mechanical. Tape precision will also help you tell you how long your fragment size is, tell you the distribution of your fragment size. So, and the quality and everything. So I think I've used qubits and I've used qubit and I've used tape precision before. I've not used nanodrop. I just have seen it. But all of them, they work perfectly in the process of DNA quantification and normalization. Again, why is this non negotiable? The amount of input DNA correlates with the final sequencing yield. So library kits have very specific input range, and too much or too little DNA will result in a failed library preparation. And of course, library preparation kits are stupidly expensive. You don't want to waste them. So that is one of the things that happen. Now I'm telling you all of this again to remind you that these are places where problems can happen. So that when you have problem when you are sequencing, you can easily think back to where do you think and troubleshoot where the problem is happening or where the problem has started from. So let's go ahead. Now, this is the next step, sequencing library preparation. And I want all of us to pay very, very important attention at this point. Very important attention. Now, I have only used two sequencing technologies in my life, and I'll be glad to explain maybe one of the two of them. Have we heard of, okay, I've mentioned it. I've mentioned Illumina before, right? And then there is Oxford nanopore technology. So these two, nanopore, don't mind my spelling, nanopore. So these two technologies have different ways of sequencing. Now, what does library prep does? Library preparation, these are two technologies, like two machines that you can use, two different technologies that you can use to sequence. What does library preparation do for you? What does it do for you? What library preparation do for you is just to help you prepare your libraries. When I mean libraries, your DNAs are not libraries. Prepare your DNA in such a way that they will be compatible with either Illumina or nanopore. You'll understand what I mean very soon. Now, Illumina, if you have heard Illumina, they are called short reads. This is a short read. They give you reads in short reads, and you understand what short reads mean. By the way, just hope and listen. And by the way, you can write these things down. You understand them. Gemini and ChatGPT will explain to you better. This one is for long reads. Now, if I want to sequence, and let's assume my sequencing is for Illumina. That means I want to do short read sequencing. I must prepare my libraries in such a way that every DNA that I'm going to be loading onto my Illumina are short and not long. Because if they are long, my Illumina will not read them, or it will read the part it can read and leave the rest to God. Does it make sense to you? It's lazy. Illumina does not have time. Illumina, I think one of the is 300 base. There is 300 base kits, and there's even 40 base kits. There's 100 base kits. So maximum is 300. They are using the kit for 300 base kits. If you should do anything more than that, you're on your own. Does it make sense to us? So when you want to prepare your libraries for Illumina, you must make sure that your fragments, right, are, depending on the kit you are using, between 150 to I think 450. Let me just use 500 base pairs long. So when you are preparing your libraries, make sure they are of this fragment. Now, for long read, I will explain this thing. Just hold on to me. Just listen to me. As the name long, so the thing long. Long read is like this. Long read is theoretically unlimited. If you are able to extract your DNA in such a way that you have 10,000 base pairs, right? If you have 10,000 base pairs in length of your DNA, good for you. In nanopore will help you sequence it. So let's now explain what this base pairs actually, what do they mean in the first place? Illumina sequence by something they call sequencing by synthesis, right? And what does that mean? It's like the material that people give you explains this thing, by the way. So if it's like when you want to replicate a DNA, or no, if you want to do PCR, how many of us here actually understand PCR? Do we know PCR or have we read about PCR? I wanna Victoria. Have you done PCR before?
I've not done it, but I have an idea.
Okay, okay, thank you. So I think most of us should read about PCR. So
I've,
PCR
I've,
is we are trying to
okay, I've
we are
done
trying
it
to
before.
oh good. That's nice. So what PCR is, you are trying to make copies of something, right? You put a primer and then you start your polymerase to start elongating the primer, by the way. So instead of doing that in sequencing by synthesis, instead of you elongating the primers, you are reading what you want to add. You know, PCR will look at, okay, this is T, I'll add A. This is G, I'll add C. This is T, I'll add A. And that, like that. But in Illumina, instead of you doing adding, if you're adding A when you see T, or adding G when you see C, adding C when you see G, you'll be reading, okay, this is C that I've seen. Oh yeah, Akọ̀wé, write C. You go to the next base. This is G, this is G that I'm seeing. There is G here. Oh yeah, Akọ̀wé, write G. There is T here. Akọ̀wé, write T. So it will be telling you what and what to be high. It's going to be, this is going to be synthesizing, by the way. It will be adding new strands, new molecule to it, by the way. And as it's adding the DNA, as it's adding the strands to it, it's writing it down. So it is synthesizing the DNA and it is sequencing it that way. So you are getting the sequence by synthesizing a new DNA strand. So by polymerizing it, you are synthesizing the DNA and you are writing your sequences. So it's very, very, very simple. Like nanopore. Nanopore is hold on. Nanopore is from the word nanopore. It has pores that are tiny. So let me start like this. There is a flow cell, and on this flow cell, there are different pores. So on this flow cell, there are different pores, like this. So these pores, don't mind me, I just draw them big. They are ridiculously tiny. So they're like millions and millions of pores. Not millions, actually. They are just, they're about 2,500 pores or so on a standard flow cell. So these pores have electronic charge inside of them. So your DNAs will just pass through each of the pores. And when your DNA pass through the pores, then the electronic charge inside the pore will change. So let's say there's 100 volts that is going through inside this pore. So let's assume, let me write something now. There's 100 millivolts going through inside this pore. If A should pass through, because A is, let's say A is horrible, but A is not as big as B, or A is not as big as T. So if A should pass through this pore, it will drop the millivolts to, it might drop the millivolts to, let's say 70. It will drop millivolts to 70. So if the flow cell will now tell the machine that my what's the name? My millivolt in this port just dropped to 70. Right, A is A that has millivolt of 70. Then if another base should go in through, say that base is T, and T drops the millivolt of the port into, let's say, 30. See, there's a big one that's entered though. T and my millivolt dropped to 30. That's T. That's T. So if another one comes in, let's say G is just 10. G should come in and pass through. Maybe just G just drop 10. So 90 kilo. So my vote just dropped by 10. So I have 90. So it's G that's entered. So that is what it keeps doing. And this thing happens about 450 bases per second. So 450 bases pass through each pulse per second. So if you have a 10,000 base pair, 10,000 base pair long read, it will go through this, and each of the base will change and alter the volt size, the voltage inside each of the ports, and that will be read into the machine, and then that will give you your sequences, by the way. So it is very, very straightforward and simple. So you can get as long possible. If you have your sequence, your DNA as long as 100,000 base pairs, this machine will read it, and you don't have problem at all. But here, you have a flow cell for Illumina. You just have a flow cell, and your strands, they will drop. Your DNA, your DNAs, they will drop on the flow cell. So when they drop on the flow cell, then you can now start there's something called there will be a bridge amplification and all those things. I don't want to go into that, but what I want to tell you now is I've explained the library to you, the technologies. Now let's go back and understand library preparation. So I hope you have understood how at least in very, very short way, you have understood how this works. You have understood that there is a flow cell where the sequencing happens. And on the flow cell for nanopore, there is a pore, and the whatever goes through, the DNA goes through the pore. For Illumina, there is the strand. Hold on, please. I don't know who's asking that question. There's the DNA. It will fall on the flow cell. It does not just fall on the flow cell, by the way. When it gets to the flow cell, then there will be a PCR will be done, then a cluster will be generated, and then synthesis will start. The polymerase will now start synthesizing. It's like PCR. You keep doing the PCR one after the other. But as it sees the DNA, it will tell the machine, okay, I've seen something here. I think it's A. When you write A, I will add C to it. Go to the next base. I see G. I'll add C to it, but this is G. You write it down. So it's as it's synthesizing, it's sequencing also at the same time. Don't mind my grammar at this point. I'm just being passionate. I want you people to understand. Somebody was raising their hand. Okay, Bola. But can you ask a question?
Yes. Good afternoon. Yes,
Yeah, good
so my question is, these two technologies that you've explained now, is there a particular one that is better than the other, and is there a reason for that?
Okay, so no one is better than the other. Anyway, most people have said that, but it is established, it is known that if you want high fidelity data that you can almost trust your life on, you go straight to Illumina. Illumina does sequencing by synthesis because it's like PCR. It will make sure the base is what it is because it wants to add something to it. So if it is A, to add T. You cannot add G to A because it is synthesizing, it will not bind. So it is very, very accurate, highly accurate, unlike nanopore. Because nanopore, if the whatever is if the pore should drop, I told you going 450 bases per second. So if it sees B and it sees A and then T bind, so it will tell you that okay, A entered though, drop 70, T has entered 2, drop 10. So it is going to be because of the speed that is going, the accuracy is debatable, even though the newer versions now are very, very accurate, highly accurate, right? But aside the accuracy that people talk about, that we all know about in science, your question also borders. If you want to work on plasmids, let me talk with plasmids. Plasmids are like extra DNA. They are DNA fragments that are not in your chromosome of your organism, by the way, or your nuclear material. So if you fragment your samples too much and do short read sequence, because if you are doing your lab preparation short read, you would definitely have to fragment. You might fragment your plasmid because your plasmid might just be 100 base pairs long. So you don't fragment them again, you will not get a complete assembly of your plasmid. So instead of that, you want something that will not fragment in your library preparation, and it will have a long read that will pass through. Does it make sense to you? So in cases like that, you want to go ahead and use Illumina if you have a question that needs to be answered with plasmids or all these mobile genetic elements. But if you don't have that in mind, please just go ahead and use nanopore. So it depends on the question you're trying to answer. That's just the whole summary I'm trying to tell you. Does it make sense?
Yes, thank you.
You're welcome. Victoria, you raised your hand. Okay, I think that was a mistake. Okay, let's go ahead. I've explained this.
Oh,
By
so
the way,
sorry, I didn't know I
this
was
is not
muted. Can you hear me?
okay. Yeah, I can hear you. I
Hello.
can hear you now. I can hear you. I can hear you.
Okay. I actually do have a question. So for Illumina, so you said that
Yeah.
it synthesizes, it produces fluid by synthesis using PCR mechanism. So I want to know the strand that we get from using Illumina, is it like a complementary? Is it like a complementary strand?
Yeah, it should be, but the technology has a way of actually giving you back the actual read. Even if it's
Okay.
complementary, you have. By the way, let me tell you something that happens. For every strand that you synthesize, we do forward and reverse read. So for for a strand that drops on the flow cell, there's something they call bridge amplification. So this, I don't want to go into details because I don't want to confuse you like yesterday. That's why I'm trying to cut it at bridge like this. So it will give you the forward read, it will give you the reverse read. So it will give you the actual read, it will give you complementary read. It's not just the complement that you get. Does it make sense to you? So it
Yes,
makes
thank
two
you.
copies. One DNA drops on the flow cell. It makes two copies of the one that drops on the flow cell. So one will give you the forward read, one will give you the reverse read. When you are getting your answer, your files in FASTQ on your machine after your sequencing, you get two reads per DNA strand fragment. Do you understand?
Yes, sir.
All right. So let me continue. Now I have explained the technology because if I do not explain technology, you will not understand library preparation. So let's go back to library preparation now. So depending on the technology you want to use, your library preparation is very, very much must be in par, must be at the same level, must agree with your like what's the name? With your sequencing technology. So if I am going to be using Illumina that is short reads. By the way, before I go ahead, when I mean short reads, I mean that this thing is just 150 to 500 p. Let's say this is 5. If I'm using a kit that is 300 base pair long, 300 base pair kit to sequence, so it will read, our PCR now, it will keep reading each of them. Once it gets to the 300 base here, it will stop. Come. If the read is as long till eternity, once it gets to 300 base pair at this point, it will stop reading. Does it make sense to you? So if you want to prepare your library, make sure that all the reads you are giving, majority of them are the read size of your fragments. Like they are at the size of your fragments. You don't want something that is longer, you don't want something that is shorter, or else you will not get anything. If you have shorter, you will get adapters instead. I will explain what adapters are very soon. But you don't want that to happen to you. So just be sure that you have everything you need. Be sure that you prepare your libraries in such a way that it agrees with what you want to do. So going on, first thing that happens. So first thing that happens is this. Let me use example of Illumina. After you have gotten your DNA, of course you fragmented them, or you might not have fragmented them during your extraction. You fragment them by the maybe not much. Because you want to work with a specific base pair, you must refragment your DNA. Does it make sense to you? So the DNA is broken into smaller pieces, mechanically or enzymatically. After you have broken your DNA into smaller pieces, you had adapters. So this is what I'm talking about. What are adapters? Because if you don't know adapter, it will haunt you when you are doing your FASTQ. So adapters are like, I'm going to slide 13. Okay, I'm going to slide 11. I'm on slide 6. Hey, Jesus. Adapters they are short DNA fragments, right? So let me use this thing. Remember, I told you that there's a flow cell that the sequencing happens. Sequencing happens on the flow cell. Don't forget that. Either Illumina or nanopore. Sequencing happens on the flow cell. Now I have this here. Let me close this one. I have this here, right? This is my flow cell. On every flow cells are not just virgin. They are not there like that. There are some complementary strands on the flow cell, like adapters. Remember that A will always bind to C, right? And G will always bind to C. If you remember that, you can make adapters here that let's say you can make, they are like maybe 12 to 18 base pairs. I'm not even sure they're up to that, right? So you make adapter strands on the DNA. So you make short strands, short strands on the DNA. On the what's the name? On the flow cell, so that you would add a complementary strand to your DNA reads. So when you have added a complementary strand to your DNA reads, they will now bind to each other, and then sequencing would happen because you are adding the library that you make. By the way, it is liquid. And if you know something about liquid, the fact that most of them are, if you are not enzymatically engineering something, you would have problem moving your adapters to your flow cell, right? So adapters are complementary strands to the strands that they are like strands that complement the uh, alternative strands that are on the flow cell. So there's a way I wrote it here. So they are small known DNA sequences attached to the ends of fragments. These acts as handlers for the sequencers. So they help you, they are like Alarino, if you are a Yoruba person. In the olden days, before now that everybody is doing boyfriend, girlfriend, I don't know you people, you sinners. The olden days, there will be someone in the middle man that will join the man and the wife that will be make them friends or something. That's the person you will speak to that will help you and find some. So that's what adapters are. Adapters are like small fragments. You would add them to your sequence, but they are already on your flow cell, so you don't have to bother. If you buy flow cell from Illumina, you must buy an adapter that would bind to your to bind to your DNA that is complementary to the adapter that is already on the flow cell. Do you understand what I'm trying to say? I hope I'm not confusing you. So for every, if you are if somebody is asking questions, I want to be sure we are on the same page. Okay, so adapters are also like your adapter that you exactly. Thank you, Abib. But let's go ask your question, please.
Okay, for the adapter, I don't like quite understand. You said the sequencer comes with an adapter. Like, can you like
Okay, hold
explain
on.
that part again? I
I'll explain
didn't.
to you. So you, you have you have um adapter allow your DNA to bind to flow cell. By the way, so you have on your flow cell. Let me draw something. Where's my scarlet drop? I cannot find my pen this way. So let me draw a line. So instead of using this is my flow cell from the side. I'm looking. This is flow cell from the top. I'm looking at my flow cell from the top. That is. If I'm looking for my flow cell from the side, this is it, right? So if you buy a new flow cell, their adapters are there already. Like they're like a socket that you want to plug to. The same way you have your sockets when you buy your things, when you buy something. So just hold on with me. They are like strand, they are waiting, they are on the flow cell. They're always there like that on the flow cell. Like this. So you would see adapters are on the flow cell. They are like this. They stand on the flow cell. So and they are like you know them, they have a node sequence already. So let's say this adapter that we have here, it has a sequence of ATGT, ATG, TGTTT, right? Now, when you're preparing your library, after you have fragmented, you must add an adapter that is complementary to this so that your DNA strand will not just be floating in the water. It will actually come to the adapter that does the sequencing. So there is an adapter fluid that you'd add to your sample. The adapter is like a motor driver. It will drive it to come and bind to this because, of course, they want to come and pack at their station, right? So you would have an adapter sequence that you would buy when you are buying your sequencing kit. It will be complementary. So you will have T, A, G, C. There should be A here. T, A, G, T, A, G, G, C, A, G, A, G, C, G, A. So you have something like this that you will buy from your vendors. And when you buy them from your vendors, you would add them to your DNA strands. These fragments that you had, right? You would add them to the end of your DNA strands. You had one at the forward there, one at the reverse here. So that if you use front, touch the adapter, you will bind. If you use back, touch the adapter, you will bind. So this is what comes. I'm trying to see if I can turn this thing. This is one. So Yeah, I really wish. Okay, I can turn it from here. Yeah, good. So this is what complements and binds to your DNA strand like this, and then sequencing happens. Do you understand?
Yes.
Good,
Thank you.
thank you. So this is adapter. They help you bind your DNA to cell. So Victor, please ask your question.
All right. Good evening, sir. So my question is this. So if there is a wrong, like if it does not bind to the sequence of DNA, it means that the flow cell
Yes.
will not be able to read it when
Yes,
doing sequencing. Okay.
it's as simple as that.
Thank
Please
you.
give me 1 minute to stop presenting. So if you what I was saying earlier is if you don't have if your adapter did not bind to your sample, it is as simple as it will not print and it will not hold on. No, no. It will not. Thank you. I was a little distracted from there. Okay, now I'm back. Thank you. Yeah. All right. Let me share my screen again. So if adapters do not bind, you will not sequence. Your DNA will just be floating in the water, be floating, and nothing would happen. That's why adapters are very, very interesting. They're like very important. Everybody, no matter the sequencing technology you use, Illumina, Nanopore, Pack Bio, there is an adapter. I think the only one that don't use adapter is Sanga. And the person that asked Sanga's question yesterday, I don't know which year we are from. Maybe we are from 1960s or 1970s. Because now everybody, no matter how little or small the laboratory is, they use next generation sequencing and either Illumina, Nanopore, and really PacBio. I've not seen them use PacBio before, though. But that's what happens. So who is asking another question? Okay, Sanga is just for gene sequencing, not for the whole genome. So it's very, very hard for you to see them use Sanga for whole genome and whatever. You will suffer. It's a lot. So continuing, after you have added your adapter, then you index. You would ask me what is indexing again, no? Right? Indexes are like unique barcodes. Now, let me explain this. I want everybody to pay attention, please. Rapt attention to everything I'm saying. Pay attention to what I'm saying. You would understand. Just pay attention. I want to make sure everybody gets this part. If you get this part, then my job here today, I'm happy. Remember, I told you earlier that when I extract, I don't extract just one sample. I extract 20 samples together, right? And when I extract the 20 samples together, I make sure that when I sequence, I don't sequence one sample. I sequence the 20 together at the same time. That's what I want you to know. So these are sample vials, by the way. So just manage them. 1, 2, 3, 4, 5. So this is 10 samples. Now, when I want to sequence my samples, I can use one flow cell like this. This one flow cell. I can use this one flow cell to sequence 10 samples together. Together, I will not like I'll finish one sample, then load another sample, then load another sample. I will not do that separately. I'll use one flow cell to sequence 10 samples. And how does that happen? What happens, right? This is what happens. After adding barcode to individual samples, so this sample here, after fragmenting, I've done my fragmentation. You know, if I fragment it, let's say this is a human being. Let's say this is, let's use human A, or let me just use somebody's name on this. Who has been asking questions? Sir? Let me use. Please pay attention to this. I would like you to pay attention to this so that we can move on. So one of these samples is here, right? So this person has a genome that is this long, right? This is the complete genome of this page of this sample. If I'm done fragmenting, I'll have this thing in bits. So I'll have them fragmented like this. And so I'll have them fragmented like this in small bits because I've fragmented them. Do you understand? Do you understand to this point? So I wouldn't have one long genome anymore. I'll have fragments because I fragmented the genome. And if it is to be a human being, I fragmented, let's say this is the gene that was for the head, this is for the neck, this is from the shoulders, this is for the leg, this is for the genitals, this is for the mammary glands, this is from gluteus, like different parts. I've fragmented the whole genome into small bits, and I don't know, it's just at random, right? So this is done for this sample. This thing also, I will do it for all the other samples like that because I'm making my libraries together. So this is sample A. I'll do sample B. I'll do sample C. On and on like that till I get all these 20 samples that I've highlighted here, right? So in my library preparation, the first thing I did was to fragment. I fragmented them. This is what you have seen. After fragmenting, I will do adapter ligation. Individually, I'll add adapter. So if I'm adding adapter, what you have here, just hold on for me. I'll have this here now. So I've added my adapter. So I've added my adapter. So this sample will have this adapter like that. All these things will be inside like that. Do we understand? Up to this point, though.
Yes.
Good. So this is for sample 1. So the same thing will happen for these other samples. DD. That is D. D. So the same thing will happen for other samples like this. I'm still treating them individually. But remember, I want to sequence them on one flow cell. All of them must be on one flow cell. If I should put all of these samples together like this into one flow cell, sure, you know that there will be problem. Because these samples have the same barcode. Let's say, let's assume this is E. coli, this is E. coli, this is E. coli. Then they would also have something similar, and then you would have issues. But if you want to, I think someone mentioned, someone raised their hand. Let me see. Victor, please ask your question.
All right. Thank you. So my question is this: how do you know the amount of fragments that a particular sample would break into, and then inform the amount of adapters that you use to ligate?
You remember the first time I told you must pull your samples together. Now each sequencing kit wants you to have a certain amount or certain concentration of DNA to start with, right? So if they have given you that when you want to start with a DNA, if you want to, one sample must be 0.4 molar concentration, one sample must be 4 nanomolar concentration. They will give you the concentration that you want, and you can calculate that with the size that you get and everything that you get, the fragment size and the concentration that you get. You know, I told you have to quantify. After you have quantified, you get your nanomole with a 1 or 2 calculations. After you have done that calculation, you will know the starting concentration. So the protocol that you are going to be using to sequence will tell you what particular point to add, what to add, and sometimes you will do calculation. They will tell you to do qubits, and after you have done your qubit, or they will tell you to measure the volume. After you measure the volume, add 40% volume of this concentration of this adapter into you. They will tell you what particular thing to add. It's based on your starting concentration. Does it make sense to you? It's from your starting concentration that you would have 1%.
Okay, all right.
Bola, to your question, please.
Yeah, thank you. Sorry. I want to ask, the adapter, is it specific or
It's general. So the same adapter you have to everybody.
okay? Okay, thank you.
Yeah. So let's continue with my illustration. I want to make sure everybody understands this thing. So I told you that this flow cell that I'm using. Hold on, please. I want to make sure that one makes sense. Sorry, just you didn't see this email this file. Sorry. So back to my work. So I have had this, and I have gotten my DNA. I've had my adapter. Now I'm going to use one flow cell. This flow cell. to sequence all the samples. Now, to sequence all of these samples, one flow cell is stupid expensive. I don't know. The last time I checked, I think the Illumina's flow cell is about 2.534 million. And I must use that one flow cell for 40 samples, for these 24 samples that I want to work with. I cannot afford to use one flow cell per sample because why? So what I will do is I'll pull up the samples, I'll put them together. And when you want to pull samples together, what happens is this. Because you didn't put any name to it. This is the head of, let's call this Bola. Let's call this Akintayo. Let's call this, let me see whose name is there again. I can use Victor. Good. Victor. So this is the gene for the head of Bola, and this is the gene for the leg of Tayo. This is the gene for, so every gene we mix together. We don't want that to happen, right? That's where indexing comes into play. What does indexing do? Indexing is also like it's a unique DNA fragment, just like we have this unique DNA fragment for adapter, but you have them plenty. So you're indexing them. If you see them, if you have seen the kits before, it comes plenty. So if you are going to be sequencing 24 samples together at once, so let's say you want to sequence 24 samples together, you would have 24 unique sequences, right? So sorry, I got distracted a little. So you want individual barcodes. So when you have seen them if you have seen the kits before, if you have not seen it, don't worry. It's like it's a well of kits, and it's a big well like that, and then you have different like 24. If you are using 24 kits, if you are going to be sequencing 24 samples together, you have 24 wells with 24 unique, like they are like primers. Do you understand? They are primers that you add to your DNAs. So you have 24 unique primers for 24 unique samples. So here we have 3 samples here. So you would have, so they are also like what would I call it? Primers. So let me use an example. After I have, this is for Bola. If I'm done adding my index for Bola would be, let's say this. By the way, so I'm just calling this in from my head, don't mind, so don't mind me. So when I had my what's the name? When I had my DNA, my index, it will be like this. There will be one here, control D, then there will be the other one here. And indexes are unique. So it's like you trying to mix. If you have been to the airport and you want to fly together, they will pack everybody's load together. But before you check in, they have tag on each of your load. So if you are checking in 10 bags, your 10 bags will have tag that are unique for you. And they will pour everything together in the cargo plane. But when it lands, you that will go and pick it, you will go and pick each of your bags. Because you know your bag, you know your tag, even if you don't know it. There was a time, the last time I was flying, I forgot, I don't know what bag, I don't know how the bag looks like because someone gave me back to fly for them. But because I know my name is on the tag, so all I did was for every bag that comes out of the chain, I looked at the tag and looked, okay, my name is not here. Until I found the tag that has my name on it. Do we get? So every sample would have a unique tag to them on the right and on the left. So after this this process, this is what you would get. Let me do that. So this is what you would get, and this is all of this that is here will become all of this. So everything will be tagged. So if you have everything tagged, you have tagged our leg, our neck, our hands, our shoulders, our legs, everything has been tagged. All the genes that are responsible, all the DNA that you have fragmented have been tagged. The same secretary will be flagged with a different set of barcode. So barcode 1, barcode 2, barcode 3 for this sample, barcode 1, barcode 2, barcode 3, barcode 4, barcode 4, barcode 5. So the thing that unites them is the adapter. But after the adapter, they have different barcodes. So after you have added the barcodes to them, then you can pull together and then sequence on is what's the name on the flow cell. So that is the whole idea of library preparation. Depending on your, so this is, if you are working with nanopore, by the way, this is the end. After you have put together, just go ahead and load on your flow cell. But if you are working on Illumina, nanopore, just go ahead. If you are working on Illumina, remember I told you about the size thingy. So you go ahead and do size selection. So size selection is, they are going to be using some things called ampere bit to select the size that you want. So size that are longer, you discard. Size that are very short, you discard. Just one size that are between the range of your kit. If you are using the 300 kit, cycle kit, you are using 600 cycle kit, you use the kit. If you are using 300 cycle kit, that's 150 base pairs, forward and reverse. Then you go ahead and make sure that your sequences are about between 150 to, or between 100 to 250 or 300 base pairs long. So these are the things you must actually do in library preparation. So all of these are very important, because if you don't do this, you are going to just be wasting your time. There's no sequencing if without library preparation, by the way. So it's just as simple as that. So let me read what is on my slide. I think I've explained everything. Fragmentation, DNA is broken into pieces, and then you had your adapters to them. So I've added your adapter, you index. I told you what indexing is. Unique barcode is added to each sample so that you can multiplex them together. You can pull them together and sequence on a flow cell. So the sequencer is designed to read DNA prepared in very specific format. Without a proper library, the instruments will not recognize the DNA. I told you earlier, the adapter must be there. A lot of other things will be added, but I just made a very brief introduction to what library preparation is. I do this on a daily basis, so I can give you a more in-depth and thorough explanation if you want me to do. But for the concept of what we want to do, you are not going to be working on the bench, by the way. So I'm explaining this to you as much as possible. So after you have done with your library preparation, then you go ahead and sequence. This is an Illumina MySec. If you have seen this before, I'm supposed to add nanopore machine to, like, I don't know why I forgot to add nanopore, because I use nanopore every day. So I told you Illumina are short reads, 150 to 300 base pairs, depending on your kit. And it's as simple. If you read what I wrote on my slide, it's high accuracy, great for finding small reads. Typically, reads are between 150 to 300 base pairs. And analogy, it's like taking thousands of very clear, detailed close-up photos of a textbook. So, of course, the DNA is the book, but you have chopped it into different sizes. You have tore out different pages, and then you are taking concise picture of each of the pages, front and back, front and back. And then after you have done that, you get your short reads, then you can align them together. Now we'll go ahead to alignment, but just hold on with me. Then we have long reads. Long reads is better for just resolving large structural changes and assembling complex. You can read as much as 10,000 base pairs. So in long read, we are taking photo of the entire paragraph of pages at once. Like, less detail in each letter, but you can see the overall structure. So the output for both of these samples is FASTQ. If you use short read, if you use long read, the output you get is FASTQ. They are intermediate files, by the way. I think for Illumina, there is an intermediate file called BCL, the binary base call file or something, BCL and then you use a tool called BCL to FASTQ to convert it to FASTQ. But that is not your business. The machine does that. The machine gives you a FASTQ file. For nanopore, there is something they call pod 5. You get the pod 5 format. It's not your business. And then you get the only thing you get at the end of the sequencing is the FASTQ. When the scientists do everything, what you need is they will get you on the server, and on your server you get the DNA, you get the reads in FASTQ, and then you go ahead and do that. I want to be sure we understand everything to this point. Do we? Because we have spent a lot of time on. Please, do we understand?
Yes.
Thank you. Thank you for answering. God bless you. You people should answer, please. Yeah. All right. I'm going to round up soon. Please just bear with me. So after your genome sequencing, what you need to do next? Okay, I've explained all of this, by the way. You have fragmentation of DNA. After fragmentation, your adapter ligation. But one thing I didn't talk about is before you add a adapter, your DNA, they don't fragment at the same way. You want to fragment them so that they are the same way, they are the same size, so that other things can join to them. It's like if you have played Lego brick before. So your Lego brick is, a Lego will not bind on top of another Lego if the cubes don't fit into each other. So you want all your DNAs to have a very good place so that your cube of adapters will bind on them. And then that's what this is. You can read about this. There are a lot of videos on YouTube. I will explain what I just explained to you in details. So I also learned it on YouTube before I worked on the bench. So let's keep that. Now we have gotten our DNA's result, and we get them in FASTQ, right? After I've gotten your FASTQ, what you want to do next is you want to do your QC and read your results. Now, your FASTQ result that comes out, I forgot to say, but it's in the, I skip it because in the manual that you are going to be, that Abib is going to give you, discuss a little bit about FASTQ. FASTQ, you know, something in sciences is we don't like to take guess. We can be probabilistical, but we want to be sure the level of confidence, right? So for every base that the software uses to call for you, if it says this is A, this is T, this is G, this is D, whatever it's to tell you. So for every base this machine gives you, let's assume this is my read, or no, let me just call AT, CG, TG, CG, AG, TP. No, that's for every base that you get, right? For every base that you get here, I want to be, there is a level of confidence that must be attached to it. That's what this second line is for. So there's a level of confidence. So instead of you getting A here, there's something we call Fred score. So to tell you J, or to tell you this, or to tell you this, or to tell you this. So you will see different symbols. These symbols are not just jargons, by the way. They are actual Fred scores. They are quality scores. They call them quality scores. So over time, you understand what they mean. But FASTQs come in four. Let me see if I can get the FASTQ file. Why did I say GASQ? What am I saying GASQ for? Yeah. So this is how it comes. You would see your sequencing ID for new entries. Then these are your sequences. Then these are this line, this port concatenates your read with your quality scores. So these are quality scores. So G, G, E, F. These things are not just jargons. They have meaning. There's something they call Fred score. So in the paper, you would understand when Abib sends the paper to you. Please make sure it's 14 pages, but it is very sweet to read. I read it very, very quickly. It's very, very sweet to read. It's 14 pages and it is nice for you to. I think you should read it. So FASTQs are reads with quality scores, but as you can see, you have a lot of things here, and these files, they are heavy. FASTQ files are very, very heavy because, of course, you have like sometimes you have like a million reads. Imagine when you fragment a DNA of E. coli. E. coli has 5.1 million bases, and let's say in the sample that you extracted from, there are like a billion E. coli there. So you have fragmented E. coli 5.1 million bases that from 1 billion samples, 1 billion E. coli samples into, so you have millions and millions of reads when you are done extracting, right? Or sometimes hundreds of thousands of reads. When you have all of these hundreds of thousands of reads, imagine having your FASTQ file for a read that is 200,000. There is 200,000 reads inside the file. You would see it is heavy. You see 2 gig, 3 gig, 16 gigabytes, 20 gigabytes. I've seen a file after sequencing, you have 16 gigabytes, 17 gigabytes, 20 gigabytes, right? Just one sequencing run. Oh, imagine if you do that on a daily basis, you will run out of space. So there are other methods. There are ways you can compress. So over time you understand how to compress these files and then how to work with compressed FASTQ files and all of this stuff. So it is not a problem there, but I'm just telling you how the FASTQ file looks like, right? Because it is important for the next thing I'm going to explain to you. Now, let's continue. What are reads? The digital output from the sequencer, short fragments of your genome. So I told you earlier, these things that I don't fragment, that I don't label. So after it is done sequencing, what I get from it is a read. So this is let's say this is from this, right? This is a read. But FASTQs don't give you just, they give you two reads. They are called paired reads. You get a forward read and a reverse read. So this sequence, this strand that I've dropped inside this flow cell, right? Or if I drop it on the flow cell, it will read it from back to right. It's also read from right to the back again. So if it's done reading from back to right and right to the left, it gives you two reads per strand. So imagine if you have 1 million strands here and you have sequenced the 1 million strand, you have 2 million reads. The forward read for the 1 million and the reverse read for the same 1 million strand. And imagine having that for 20 samples or 24 samples that you run together. That's a lot of reads that you have, right? So having to go that. So those are just that's read so that you don't get confused when somebody says when somebody mentions reads in sequencing. That's one of the basic things you must first know what are reads. And now I told you what reads are. Then what is QC? So assessing the raw reads to ensure high quality before analysis. We are checking and removing low quality bases. So what are low quality bases? They are bases that when you understand Fred score, you understand low quality bases. There are some bases that you don't want to see. When you start seeing all those symbols, this question mark, this is equal to, the reads are of low quality. If you read Fred score, don't let me put anything in your mind. Read Fred score and you understand fast skills. I think after today we should start giving assignments, by the way. Read Fred scores, you understand what fast skills are, by the way. So if you have low quality reads in your file, you want to remove them. If you have adapter, I remember I told you adapter. All my explanation for adapter is for this point though. After you have done with your sequencing, and adapters were added during your library preparation, they are not part of your sequences. So you want to remove your adapters, you must remove. You have to remove adapters. By default, it should be removed, but sometimes they are just interwound into your DNA sequences. So your adapters must be removed, and then you can go ahead. And of course, if you have contaminant DNA, one way or way, you must remove all of these things. So you are checking and removing all of these things from your sequence. How do we do it? We use FastQC. Is a number one to everybody knows FastQC. It's sorry for short reads. Everybody use FastQC to get the quality metrics of their data. And then if you are using Kraken or Bracken, you are using it to check and to identify contaminating species. We also use this for metagenomic DNA, by the way. We'll use this here too. And trimming tools, Trimomatic, you have to use it to filter bad data. So if you have data, all those data that have, you can tell you, it will tell you Trimomatic that any data that is less than 20 quality score, you understand the reason why I said 20 data when you start reading Fred scores. After you have read the Fred scores, after you tell Trimomatic, any that is of low quality, remove it. So the reads that you have will now be reads of very high quality, and then you can go ahead and do. Your analysis is dependent on your reads. If you have good quality reads, you have good quality results. If you have bad quality reads, you have bad quality results. Do you understand? So moving on. After you have your read, then you do genome assembly. I told you have different reads. You have like 1,000,001 reads. So assembly is you trying to, I told you earlier, we have a complete genome, then we fragmented it, then we sequenced. But now you want to make that reference, you want to complete that thing that you fragmented in the first place. You want to fold it back. You want to make the complete genome that you have fragmented back. How do you get it? All those puzzles that you broke, all those reads that you have broken down into pieces that you have sequenced, you must stack them. It's like puzzle. You must stack them back over each other again. And how do you do that? You can do it two ways. The first way you can do it is to have this reference genome. If you know that this organism that you sequence was E. coli, you can just get a reference E. coli from the NCBI database. And then all your reads, you now align it towards that read. So you align them side by side. After you are done aligning side by side, you now say, okay, this one fits to this part, this one fits to this part, this one fits to this part. Then you start aligning and arranging. And at the end, you get something that is almost complete, or sometimes, most of the time, it should be complete, sometimes not complete, but you will get something that is better, that is more complete than the read that you have. The reads are just fragments. So, and there are different stages to this thing. And that's reference-based assembly. I explained that. There's also de novo assembly. De novo assembly is you are not really using a reference. They will arrange themselves. So you start looking for similarities. Oh, now I have ATCGT. The second is okay, I have CTG. So we can complement each other. Then we'll start arranging, and they'll start arranging like that. So there is pros and cons to this, by the way. But that's de novo assembly. Assembling genome from the scratch without using a guide. Used for new organisms. So analogy is assembling a jigsaw puzzle without the box lid. But if you are using reference assembly, you have a box lid that will guide your puzzle. So what's the output? You have contigs, and that's continuous assembled sequences. So if 1, 2, 3, 4 here, I don't know if you are seeing my mouse, I don't know if you can see. So different contigs, different reads will map together and then they will form a contig. So the longer the contigs, the better you have. Then at the end of the day, you can get a FASTA file containing your final assembled genome. So FASTA is FASTQ without the assembled FASTQ without the quality score. Does it make sense to you? So because when you're assembling, you remove the quality score. The quality score is for you to do your QC. And after I've gotten your QC done, just assemble and remove because it's heavy, it's redundant. You don't want it, right? Then you go ahead, you get your FASTA file. After you do genome assembly, you can now start doing a lot of things that you want to do. I mentioned it's genome annotation, variant calling, AMR gene detection, phylogenetics, typing, MLSA, and stereotyping. As much as possible, you have the data with you to do everything you want to do. I hope we have understood what, because listen, she mentioned everything yesterday. She explained it. I just wanted us to break. I wanted to break it down because I'm sure our accent, has thrown some people off. People were sleeping. I could imagine. I can see people sleeping in my head. That's why we had to go through this again. I hope we understand everything that we have done today. If you have questions, please ask questions. And if not, thank you. Oh Lord. Ask questions, oh. Thank you. Please, I would like you to, if you have understood the question, I would like to, if you can say think you understand, and then you can write in the comment. Just I want to be sure that people understood what we have done. Michael, you wanted to say something?
Okay, yes. When you add the adapter does it randomly pick and bind to the top and the to the beginning and the end of the little fragments?
So it does, but there are two types of. But I didn't want to get until I don't want to go to the details. It depends on if you are using Illumina. There is P5, there is P7 adapters, and these adapters pick what part to stay with. Some stay on the top, some stay at the end. Do you understand? So when they, so it depends. But for nanopore, if you had the same nanopore goes on both ends, the same adapter goes on both ends, so you don't have any problem at all.
Okay.
Yeah. So every DNA strand that is in that pool, once you pour your adapter, they will bind. That's why you have to prepare your end. I didn't stress end preparation first. After you have fragmented your DNA, if you remember science, if you remember your basic genomics, you have serrated edge, or you can have blunt edge, blunt end and serrated end. So if it is serrated end, your DNA is double helix, right?
Yes.
One edge will be longer than the other one. That's serrated edge. But if it is blunt edge, both ends will end the same way. So one will not be longer than the other, one will not be shorter than the other. So the first thing that you do after you have fragmented your DNA is what we call end preparation. And end preparation, I have it in my side. I just didn't mention it that much because I don't want to confuse people. I've done enough confusion. Here, after fragmentation, you see some that will overhang the other, some will be too long, some will be the same. But your DNA, your adapters and whatever, they want to bind. There's a way they want to bind. So if they don't bind the same way, there will be problem. So you first do end repair. And end repair is anyone that is longer than the other. You cut so that they are the same size. You are using a scissors to trim the blunt edge to be smooth. Then after you have cut the end, then you now do A peeling. You just put A there. Because your adapters that you're adding, they have a little overhang of T rather. So they have a T that will complement the A. So it's just all of these things is for the adapter to find the end of the DNA. That's all. So if you read the protocols of nanopore of Illumina, specifically Illumina, you would understand why they do these things. But it's very, very interesting. And it's science, it's basic science of A bind to T and G bind to C. Do you understand?
Yes.
Yeah. Thank you very much. Any other question? Victoria, please ask your question.
Yeah, thank you very much for that section.
Yeah.
Please, I would like you to. I didn't get the whole concept of QC. That step, I think it's the step
Okay.
six, QC and read
I
QC. I'm not so sure whether I got it.
QC and what? QC and what is that? QC and what? I didn't hear what you said well. Maybe it's my internet.
And read trimming.
Okay. Read trimming. Thank you. So let's go to the slide. I didn't mention much of QC. I think one of the things we'll first do is QCs because it's very important. So QC is quality control. In science, I don't know what department you are from, but if you do any experiment, you must do a quality control. I am in the laboratory science profession. I work with patients. Where I used to work with patients. I know they work with patients again. I'm now working with bacteria. But if you want to work on it, let's say you are in chemical pathology and they ask you, maybe at least most of us have gone to do glucose test for our fathers before or something. If they want to do that test in the laboratory, they must run a control. They must run a standard. So that the figure they are giving you is sure that it's from your father. So they must validate the machine is fine, and then they must validate that the reagent are using is okay at the same time. So in science, my point is in science, everything must be assured and quality backed, right? Because that's why science is science. Science is not just religion, it's not faith based. Science is evidence based, right? So the same thing here. You want your machine to tell me that this machine, this thing is A, this thing is T, this thing is G, this thing is C, and you want it to tell me with very high degree of certainty that this AO is A, this GO is G, this CO is C, and this T is T. You don't want to tell, there are some that the machine will guess. There are some reads that when you see, there's something. Let me just. I have like there's 1 million things that are popping in my head I can't see, but I want to take it one after the other. So these are reads, right? This second row. You can see my mouse, right? Can we all see my mouse?
Yes.
Good. So this second row are the reads. This is cytosine, timing, cytosine. These are reads, right? So these reads are what the machine has given us. I guess this is Illumina, right? So if Illumina has given me these reads, then I want the Illumina machine that is giving me to tell me how sure it is. How certain are you that this read that you are giving me are actually what it is in the DNA? Do you understand? I don't want you to be guessing that at position 4, I'm guessing it is A. It might be T, it looks like T. But I'm thinking it's A. You don't want that guess. You want an actual answer. You want quality. I want you to give me the probability that you are very, very sure that this is A, this is T, this is G, and this is T. That's what this line is for. This line 4, they call them quality reads, quality scores, or some people call them Fred scores because of the person that invented it. I don't know, my channel Fred is in his name, right? But what does these things, what do they do? Let me open a new tab. Let me open a new tab and explain. So, when you see B, C, D, E, F, G, H, all those things that you are seeing, if you see them here, by the way, these things are these signs you are seeing here are from these Fred scores, by the way. So if you see all these figures, they have the higher. If you see the letters right there, rather, they have I. So from art, let me use the second line here. So, Illumina 1.8 or later. So base 33. This is what we are using now. So art A, B, C, D, E, F, G. They have highest Fred scores. So you can see their scores. The highest number of Fred score is 40. So and Fred score is the they call it the probability for error. So I didn't want to take you deep. You want to carry me deep like this, but let's go. I hope I'm able to explain to you as much as possible. Now, if I have a Fred score of 40, what it means is this. What it means is, what's the probability that you make mistake that the A that you call for me that is A? Like, let's say I. This is I now, and we say I here is 40. I here is 40. What's the probability that this A, this T that you call for me that is I, or this A that you call for me that is I, what probability that it is actually A that is in the sequence that you read? Because you just read it to me. I am hoping to believe that it is you. So the machine will tell me that my probability is I. And what I means is, if you read this thing 1 million times, 9,999 times, it will be I. It will be A. It's just that one time that you will be something else. And that's a very high level of confidence. If I tell you that if you call me 9,999 times by my name, I would answer you. But if you call me maybe by one, that level of figure gives you confidence. It's a probability of the level of error that you can call. When we get the fast kill, I think Abib or something will take this part and explain to you. But it's something those Fred scores help you, gain confidence. So if I have this I, if I have a first score of 38, so 38 might be, I can't remember how we calculate at the moment, but this is the formula that they used to. I think this is it. Oh, good. Not up here. So base accuracy is 99.99. If you have a score of 40, if you have a score of 30, your base accuracy is 99.9. So this is still good, right? But if I have a score of 20, then I'm not sure. It's 99%. So if that sample is A, 99% of the time, it is actually A. But I want something that will tell me 99.9999999. So I like figures from here. So any figure that is below 20, I don't pick. So when I'm doing my trimming, when I'm doing my QC trimming or my QC control and whatever, my trimming, I want to remove every reads that have quality score of 20 and below. And how do I know quality score of 20? See, from my, here. Okay, 20 here is, no, this is not the thing I was using. No. Where is it? Okay, here. 20 here is 5. Is that the 1.8? Yeah, I think that is it. Though. Yeah, 20 is 5. What is that thing? What? The symbols. Where did I lose this?
Image.
The first one. Oh, 33. Oh, I was using 64. Sorry, 33. So
No,
this
what was this image you had?
the first image. I couldn't find it again. No,
The,
because I knew it was
the third image now, like this. Oh, like after the one highlighted, the next yes.
Oh, okay. Look at this. Look at this.
So this the
So A is 30, right? Oh yeah. A is 31. Then if I go down to 20, then you start seeing symbols, all these Jagbajantis figures. So if I want to trim my figures, if I want to trim my reads, I want reads that I am sure are like 99% accurate. So if the machine runs that thing 100 times, 99% of the time, that base will still be the base that it called for me. So any read that is below 99, that is below 20 quality score, remove it, and that's what the trimming does. Remove quality scores that are lower than the quality I want, number one. And number two, remove quality scores. I remove adapters. If I had adapters in my work, but adapters are not part of my sequences. Adapters are just what I want to help me draw my sequence to the flow cell. So it should be very, because it should be very insane if my adapters actually now starts dominating my day. And that's one thing I skipped when I was talking. If you are fragmenting your DNA when you are doing library preparation, fragment your DNA in such a way that it's not too short and it's not too long. If it is too short, you will be sequencing adapter, you will not sequence anything. Now adapter you go to sequence. And if it is too long, you would miss a lot of your sequences. Does it make sense to us? So that's the whole idea of QC and trimming. Do you understand, Victoria?
Yes, thank you very much.
Thank you. I'm happy you understand. God, teachers, teachers are trying. So do you have any other question before I round up? Because I've spent over 2 hours here. God.
So my question is this: the poor reads, are you going to like replace them with what you think is right, or delete them completely?
You delete them completely. You delete them completely, entirely. Because you know why I have confidence of deleting them? Because there's something they call read depth. I don't want to go deep, but when you start the process, you would understand these things. You can hold this question when we start working with data rate and you understand this thing. So I'm sure that even if I delete this thing, maybe there is another segment somewhere that has sequenced it better, right? So that sequence that have sequenced it better, even though they are different, it will complement for the part that I've removed. You know, I have a million reads. Do you get it now?
Yes.
Exactly. Thank you. And I make my work show. Oh yeah. Next question before I round up, because I've finished all my juice like this. So Tochi, I think it's over to you. Nobody will ask questions, you know. Now I go sleep. Ah, Habib, please take over. Where am I, guys? Is it my network? Can you all hear me?
Yes, we can.
Yes, I
We
can
can
hear
hear
you.
So Habib and Tochi, what are you guys?
Sorry, one question just popped up in my head though.
All right, all right.
Don't yet sleep. Okay, so you told us that the sorry for the noise. The fast queues they are read with we would have
Yeah,
two strands, like the forward and the
forward and
reverse
reverse.
strand.
Yes.
So which one are we to use? Like how will you know which one is forward? How will you know which one is
You don't,
reverse?
so the machine,
And which one
the
are you
machine
working
names
with?
them. It names them R1, R2. When you start working with them, you understand. You will see them. The
Sorry.
machine. I can hear you. Can you hear me?
Okay, I can hear you now.
So the machine names them R1, R2. R1 is forward stand and R2 is reverse stand. And you don't have to bother which one do you need because you need the two every time you work. The machine will ask you for the two. I'm sorry, why am I saying machine? The software that you are using will always ask you for forward and reverse. Any tool that you will need to work with that needs you to work with fast queue, they ask you for forward and reverse. So you just load it. Thank you. I don't need Miss Yan. You are laughing at me. All right.
Hi,
Um.
good evening, sir. Sorry, I have a question, sir.
Okay.
Yes, yes. You showed us a FASTQ image, not long.
Yeah.
You mentioned that one of the lines is the read, the fourth one is the
Quality
Fred
score.
quality
Yes.
score. Yeah. So I want to ask, does the rest of the lines do they have any importance, or should I say, what are the significance? What's the
Yes.
significance of?
They do. So the first four lines are for read, are for one read. See the second four lines is still the same thing. A fast queue starts with an at. And how do you know you don't cram it? Fast queue. If you turn your queue, you can use your queue to write at. So that's how I knew that. A fast queue starts with a question, a greater than score. And if you can, how I learned that before I finally knew it is A fast A. The A you can angle it and it becomes a greater than sit down. So let's move back to fast queue. Fast queue starts with the at. Then this is the first reads on this file. So it will, when it's getting too long, it will cut it. So it is a complete read like this. But because it's getting too long, and when you start your software, you used to read it. Imagine, when we used to read our notepad, that you will see line that you will be you start scrolling to infinity. Instead of having that problem, they will cut it to the new line. So this read continues here, and it will still repeat the same thing at, and then read 42, machine this, flow cell this, and then the same read continues. Plus, it's still the same thing. Oh. So you see, it's 8, 1, 2, 3, 4. The first 4, 5 to 7, 8 is still the same thing. It just continued. Does it make sense to you?
Yes, sir. It does. Thank you, sir.
Yeah, thank you. Any more question? I guess Adib and Tochi are not. I don't know why they are not saying anything. I want them to take over so
I've
that.
been trying to talk since, but I guess my network is bad. Can you hear me now?
Yeah, we can hear you now. We are round off. Thank you.
Okay, yeah, thank you very much. So if you have more questions to ask, you can also fill the form from yesterday, and then we can get back to you later on. So and then on the general group too as well, or you can ask your mentors or your fellow peers on the WhatsApp community. So I think that's all for now. So by next week Friday, we will be taking on, I think, computer architecture, right? Yes. And after
Yeah,
that, we'll
computer.
dive into, yes, we'll dive into command line. So I'll be sharing a survey that we're going to fill. Maybe sometime just after the next week, I'll share a survey that I'm supposed to fill for the course. And I think that have a test this week. So don't worry, to be fair, just like a short test for people so far on mode one. We're going to module 2 for the computational parts. Yeah, so thank you very much, everybody. See you next week, Friday.
Bye.
Yeah, bye.